Hello, everybody. A very good morning and afternoon to everyone virtually, too. Welcome to today's webinar introducing LocIn, Bio's new targeted integration system for the efficient creation of isogenic mammalian cell lines. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and brought to you by Bio. To learn more, visit www.rukin.bio. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I would like to now welcome our speaker, Dr. Timothy London, Director of Molecular Biology and Cell Engineering at Bio. Dr. Timothy London, you may now begin your presentation.
Thank you everyone for joining us today for this webinar, where I will describe our recently developed LocIn system and its use in the efficient creation of isogenic mammalian cell lines. Before I begin, I'd like to thank LabRoots for hosting and facilitating this webinar today. Before I get into the scientific portion of my talk, I'd first like to introduce Bio and what we do. We are an immunology-focused CRO based in Scotland in the UK. We are located approximately 20 miles outside of Glasgow in a town called Motherwell, where we have our research labs. We have more than 100 employees and we are still growing. Our vision is quite simple, and that is to become the most trusted research partner for biotherapeutic innovators. In terms of our expertise, we focus on a range of therapeutic classes, from antibody-based drugs and advanced medicines, including immune cell therapies, through to nanoparticles and small molecules. We are a world leader in human immunology research service provision for the assessment of immuno-oncology, autoimmunity, and inflammation targeting therapeutics. We apply a unique combination of immunology, bioanalytical, and molecular biology expertise to establish advanced experimental solutions to assess the safety and efficacy of therapeutics. We support a global customer base who operate from the earliest phases of drug discovery all the way through to the clinic. Before talking about their applications, it's important to understand what cell lines actually are. Cell lines are populations of cells that scientists grow in the lab. Under the right condition, these cells can divide indefinitely, which makes them incredibly useful for long-term research and drug development. What's unique about cell lines is that they are usually derived from a single original cell. This means that all the cells in the line share the same genetic makeup, giving us a consistent and controllable model for experiments. Two well-known examples of cell lines include HeLa cells, the first immortal human cell line derived from cervical cancer cells, and CHO cells, which come from Chinese hamster ovaries and are widely used in biotechnology, especially for producing therapeutic proteins. Cell lines play a critical role in drug development, starting with predicting how a drug will behave in the human body. Because they model human tissues, they give us early insights into potential responses without needing to test in humans right away. Next, they're invaluable for screening drug candidates. Using cell lines, researchers can rapidly and cost-effectively test thousands of compounds to assess both safety and effectiveness, allowing us to narrow down the most promising options. They're also essential for studying the mechanisms of action. Cell lines allow us to delve into how a drug interacts with specific cellular pathways, which is crucial for understanding both therapeutic effects and potential side effects. And finally, from a regulatory standpoint, cell lines help meet early-stage safety requirements. They provide foundational safety data that's often required before progressing to animal studies or clinical trials, making them a key part of the approval pipeline. Traditional cell line generation is both time and labor intensive. It starts with the transfectional transduction of your target cell line, usually with antibiotic selection following the transfectional transduction, to generate a pool, and then that pool requires analysis to see how the target antigen of interest expresses. Usually at the pull stage, the expression profile is heterogeneous, so if a clonal homogeneous expression cell line is required, then a subcloning step is performed, usually either by single cell dilution cloning or via fluorescent activated cell sorting, or FACS. From that single cell sorting, clones are then expanded and then analyzed individually. This can be quite labor intensive in itself, depending on the number of clones that you pick to analyze. And then from that selection of clones, you pick the best one. So what is the LocIn system and how does it help with cell engineering? LocIn is a targeted integration system that allows the efficient generation of genetically identical mammalian cell lines. The important feature of this system is that the subcloning step, which I mentioned in the previous slide, is not required. So the overall cell line generation process is both simplified and shortened. You can see with the lentivirus approach that there's a 6-week period for the construct design, synthesis, and lentiviral packaging, and then 2 to 3 weeks for the pull growth and subcloning, and then another 4 weeks for the outgrowth of clones and analysis. With the lock-in system, the synthesis phase is shortened because lentiviral packaging is not required, and then the actual cell line generation process does not require the subcloning step, and when the cells grow through selection, you have your final clone at the end of the outgrowth. In the next few slides, I'm going to give you an overview of how the lock-in system works, which is based on a process called Recombinase Mediated Cassette Exchange. In order to build the lock-in system, we first integrate a landing pad construct into the cell line of interest. The landing pad consists of a GFP gene for fluorescent expression, an HSV thymidine kinase gene, which I'll come to in a second, and then a hygromycin resistant cassette. This is flanked by 2 different integration sites, which again I'll explain in a second, followed by a split blastocidin resistance cassette. We put this whole landing pad into the target cell line either via transfection or transduction to create what we call our multi-platform system, which allows for the high expression of your target gene of interest due to the fact that there are more than one platform or landing pads integrated into the genome of the cell line. We are also working on a single integration platform system, which we will put the landing pad in via CRISPR integration into a defined genomic locus. So in mammalian cells, this will be the safe harbor AAVS1 locus, and in CHOK1 cells, this will be the ROSA26 locus. The presence of the landing pads is maintained by hygromycin selection, and the EGFP expression also allows us to confirm and profile the expression from these cassettes. So we have our landing pad or platform cell lines. How do we then go about retargeting them? To achieve this, we use a retargeting plasmid, which contains your gene of interest and 2 different recombination sites that recognize the recombination motifs in the platform cell line. This also contains a split blastocidin, the N-terminal portion of the split blastocidin, that complements the C-terminal portion of the split blastocidin in the platform cell line landing pad. By co-transfecting this cargo delivery vector containing the gene of interest, along with a recombinase enzyme, a process called Recombinase Mediated Cassette Exchange occurs, or RMCE, where these 2 different recombination sites recombine with each other, which effectively flips the landing pad cassette out of the genome of the cell line and replaces it with the gene of interest carried in the cargo vector, and at the same time the split blastocidin is recombined so that you have a complete blastocidin resistance marker. This allows you to then select for a successful recombination via blastocidin selection. So what does this look like in practice? So you have your platform cell line, which expresses EGFP from the landing pads that are already integrated into the genome. The example I'm using here is the multi-platform system, and you can see denoted with the little green bars the different platforms within the genome. We transfect these cells with the donor vector carrying your gene of interest and another vector carrying the integrase which performs the recombination. After 24 to 48 hours, blastocidin selection is applied. What results from this is what we call a partially retargeted pool. Within this pool, you will have instances of where there's been retargeting of one of the platforms, perhaps some of the platforms, and then in some cases you will have cells where all of the platforms are retargeted. This is where the thymidine kinase comes in. Thymidine kinase converts ganciclovir into a toxic compound which will kill the cells. Therefore, any cells where all of the copies of the thymidine kinase have not been flipped out by the Recombinase Mediated Cassette Exchange will be killed because the thymidine kinase that's still present in the platform will convert the ganciclovir into the toxic compound. Only the cells where all of the copies of the thymidine kinase have been flipped out will survive the ganciclovir selection. So effectively, at the end of this secondary selection with ganciclovir, you will have what we call a clonal pool. All of the cells will be the same in that all of the platform or landing pad sites will be retargeted. I'll now take you through some data that we've generated on the LocIn platform system that we built initially in our prototype CHOK1 cell line. Shown here is our CHOK1 multi-platform system where we've integrated multiple copies of the EGFP expressing landing pad and then selected with hygromycin selection. We initially made a pool and then cloned these out to generate high expressing clones that we can monitor via expression of the EGFP. We also looked at stability of expression from the platforms over time in this clone. So you can see here that we have monitored GFP expression both with and without the hygromycin selection antibiotic that resides within the cassette up to about 35 days. And you can see in this graph here on the right that the expression level remains constant over the course of the 35 days. Having built our platform system, we then wanted to see how it performed after being retargeted. To achieve this, we use a donor vector that expressed EpCAM. So on the left, we started with our unretargeted platform cells, where you can see clearly the high expression of GFP and no expression of EpCAM. We retargeted them with the EpCAM plasmid and then selected with blastocidin for approximately 10 days. You can see here immediately that the EpCAM expression is now showing quite highly in the graph at the bottom, but also the GFP expression has actually reduced, and this is what we would expect given that as the recombinase mediated cassette exchange occurs, the GFP is actually flipped out of the landing pads and is lost. So you can see in the middle that the GFP expression has reduced, and there's also a population where there's no GFP expression at all. After the initial blastocidin selection, we then applied the ganciclovir selection. This, as I mentioned before, will then kill any cells where all of the landing pads have not been retargeted. So you can see after another 7 days in ganciclovir, the GFP expression has almost completely been lost, showing that the only cells remaining now are those where all of the landing pads have been recombined so that they now only contain the gene of interest, which is EpCAM. And you can see by removing the partially recombined or partially retargeted cells from the pool via the gancyclovir selection, the EpCAM expression is now higher than it was in the pool. In an earlier slide, I showed you how the platform system has stable expression from the landing pads that are integrated via monitoring the expression of eGFP. We wanted to then look, once this cell line had been retargeted with EpCAM, was the expression still stable from the retargeted landing pads? And you can see in this graph, again, we monitored expression over 35 days, but at this time we were looking at expression of the EpCAM. And over the 35 days that we've monitored, the expression level remains stable. And this is whether or not you keep the selection antibiotic, the blastocidin, present. Even in the absence of the antibiotic, the expression still remains stable. So we have shown that the LocIn system can be retargeted successfully with EpCAM, and that over the time period that we monitored, 35 days, the expression level remains stable. But we also wanted to see how it performs when we're retargeting it with a complex multi-pass transmembrane receptor. So the example that we've chosen here is GLP-1R, which is a 7-pass transmembrane GPCR. Again, as before, we start with our unretargeted platform cells. We retarget with the GLP-1R plasmid along with the recombinase expression plasmid, and you can see after approximately 11 days in blastocidin, the GFP expression has reduced, but it's not completely gone, and we now have expression of the GLP-1R as measured by flow cytometry. We then apply the gancyclovir selection. This should remove any cells where there's only been partial retargeting. This took a little bit longer than the EpCAM example, probably due to the complexity of the GLP-1R, but after another 15 days in gancyclovir selection, you can see that the GFP expression has almost completely been lost, and we now have what looks like a clonal population of cells expressing GLP-1R at quite a high level. Finally, to show that the system can work with a range of different types of target antigens, we tried retargeting our platform system with a chemokine receptor, in this case CCR8. Same as before, we start with the unretargeted platform cells, retarget with the CCR8 contained within our donor retargeting plasmid along with the integrase expression vector, and then select with blastocidin. You can see here, as before, the GFP expression drops due to the partial retargeting, and we get some expression of CCR8. Applying the additional gancyclovir selection, this time for 17 days, we have again completely got rid of the GFP expression, and we end up with a clonal pool of cells expressing the CCR8 at a high level. So in summary, the LocIn system is an advanced targeted integration platform system that allows you to efficiently generate genetically identical isogenic mammalian cell lines. The way the system works allows the removal of the need for subcloning and analysis of multiple clones, and so both the time and effort required for cell line generation is reduced. One of the important features of this system is that it actually allows for the accurate comparison between different construct designs, because the integration pattern is already preset in the platform cell line. Therefore, in all cell lines derived from this system, they all have the same integration pattern. Thus, that removes that variable when you're creating cell lines in order to compare different construct designs, for example. That brings me to the end of this webinar on the LocIn system. Thank you very much for your attention, and I'll be happy to take any questions.
All right. Thank you, Dr. Timothy London, for your very informative presentation. We will now move into the live Q&A portion of the webinar. So if you have a question that you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We will answer as many of your questions as we have time for. Okay, let's get started. The first question that we have here is, what do you see as the main advantages to employing lock-in versus conventional cell engineering approaches?
That's a great question. We believe there's a few advantages to using the lock-in system. The first is it's faster to generate the final cell line compared to the conventional approach. Um, we also believe that because of the fact that the landing pads are already integrated into the cell line, the integration pattern is the same if you're comparing different constructs used to make cell lines using the lock-in system. So you can remove the variable of effectively random integration from your cell line generation. And the third advantage is that it's simpler for making cell lines because you don't have to do the subcloning step and then grow up the individual clones and analyze them. So we think it's both a more efficient and quicker way of making cell lines where you can make precise comparisons between different construct designs.
Okay, great. Thank you. We have another question here. It says, what other cell line backgrounds will you be making?
So we're planning on putting this into at least HEC293 cells, so we have the human cell background, and we also think that GERCAT cells would be a good background to build this in, particularly if you're interested in comparing different recombinant TCR designs, for example. This could be combined with a TCR knockout GERCAT cell line. Beyond that, K562s are a possibility, but I think we'll be guided by what our customers are most interested in. But those are the plans for the sort of medium term.
All right, wonderful. Thank you. Well, thank you again, Dr. London, for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Rukin Bio, for underwriting today's educational webcast. Before we go, I would like to thank the audience for their very interesting questions. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. With that, I will actually go ahead and pause. It looks like we have two more questions that have come in here, so I'll go ahead and jump to those. This one says, are the GFP integration sites in the multi-platform system random?
They are in the multi-platform system, correct? They are. So we deliver the landing pads via lentivirus. So the integration sites are random, but they tend to be biased towards transcriptionally active regions of the genome. We also are building the targeted integration cell line where we are putting a single integration into a defined locus. So that is the ROSA26 locus in the CHOK1 cells and the AAVS1 locus in human cell lines. So yes, the multi-platform system is random, but the single integration, targeted integration system is at a defined locus.
Got it. Okay. And we have another here. It says, would it work on human diploid cell line?
Yes, yes. I mean, we've used CHOK1 as our sort of prototype, but yes, it will work in human cell lines as well, and that is the plan, to build it in human cell lines.
Okay. Wonderful. Thank you. With that, we will go ahead and close out for today. Again, thank you, Dr. Timothy London, for your time today and for your important research. Any questions that are submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everybody, take care. Goodbye.